Liger.
What's a Liger?
It's pretty much my favorite animal. It's like a lion and a tiger mixed. Bred for its skills and magic.
The liger has a gene that lets it have black stripes. The gene codes for proteins that gives the liger the trait. The liger goes through a process of transcription, which is the process of making a RNA copy from a DNA template. The start of transcription is initiation, where RNA polymerase binds to a location on the DNA called a promoter. Promoter signals the beginning of the black stripes gene. Then the gene goes through elongation, which is when the RNA polymerase makes a complementary RNA strand from one of the exposed DNA strands. mRNA has codons that code for individual amino acids. This DNA strand is called the template strand. Next, the gene goes through the third step of transcription called termination, which is when the RNA polymerase comes across a DNA sequence called a terminator and stops the transcription process. The eukaryotic mRNA transcripts must be edited. The original mRNA contains sequences known as introns that don't code for anything and exons that actually code for a protein. The introns are then cut out and the exons are spliced together. A cap sequence and a tail sequence are added and the mRNA is ready to go. The black stripes gene also goes through a process called translation, which is the process in which a ribosome reads the mRNA and makes a protein. The first step of translation is initiation, which begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. Next is elongation translation, saying the stage of met-tRNA is in the P site of the ribosomes. The next amenable tRNA enters the A site of the ribosome. Then the met-tRNA and the next amenable tRNA are now next to each other in the P and A sites. Next, the ribosomes then translocates one codon to the right in the mRNA. Last but not least is termination translation, which is when a stop codon is encountered in the A site, and a release factor enters the A site. When a stop codon is entered in the A site, a release factor enters the A site and translation is terminated. Next, a mutation occurs, which is when any kind of change to the base sequence of either DNA or RNA. When the mutation occurred, it removed a base in one amino acid after this mutation is altered, making the stripes turn from purple to black.
Baby, I'm playing on you.
The end.